Hello, everybody. I hope you can hear me just fine. For talks like this, I often like to start not with my research, but a little bit about how I got into the position I am in currently at the Lincoln Park Zoo, just because I feel that that's pretty helpful for a lot of people that are working to become quantitative ecologists themselves. So, yeah. Like Lilac said, I grew up in Washington State in lovely Puyallup, Washington. I, of course, still know the Puyallup Fair theme song, but I will not sing it for you today. But I lived there for about half my life. I did my undergrad at Western Washington University up in Bellingham in environmental science. And after that, I did absolutely nothing with my degree. I went and moved to Dublin, Ireland, and I worked as a bicycle mechanic while my girlfriend, she's now my wife, got her master's in theater there at Trinity College. I, of course, working as a bicycle mechanic and riding around and drinking beer with people, had a much more fun time than she did in a very intense one-year program. And so I have very fond memories about my time in Ireland. Well, she kind of does not, unfortunately. But after that, we decided, kind of on a whim, to move to Chicago. Job prospects back in the Pacific Northwest weren't really there, and it kind of felt like a step backward after living in Ireland. And so we just moved to Chicago without any job prospects, anything like that. Kind of taking a big risk there, you know. But I got my first job at the Lincoln Park Zoo, actually not doing any science whatsoever. I got a job at the zoo cutting animals for, or cutting, sorry, I mean, I did cut animals, but I cut food for the animals in our care at the Lincoln Park Zoo. Um, and sort of functioning as a sous chef. And while I was working there, I saw a number of people carrying camera traps and wearing binoculars and things like that. And I thought to myself, like, well, that's the stuff that like I trained for during my undergrad. Who are these people? And little did I know that there was a really large conservation and science department directly above my head, just one floor above me in the building I was working. And I found out that there was a science center there called the Urban Wildlife Institute. And those were the people that were carrying around camera traps and things like that. And I was able to switch jobs at the zoo and start working as an intern with them and moved up steadily through the ranks working at the Urban Wildlife Institute. After a few years there, my boss, Dr. Seth Magley, convinced me that it was a good idea that I should go and further my education. And so I went and started my PhD at University of Illinois at Chicago with Joel Brown. When I started my PhD, was not interested in statistics or anything like that. I liked birds. I mean, I still like birds, but that's the main reason why I started my PhD. And I realized about maybe a year in that I came in with a bunch of camera trap data, things like that to work with, and the questions that I wanted to answer, there was just no statistical models that really existed to address those questions. And so I guess just out of stubbornness, I decided that that was what my PhD was going to become. So I went a very quantitative route during my PhD. After 4 years, I wrapped up my PhD at UIC, and while I was starting to look around for jobs and things like that, the Lincoln Park Zoo said, "No, thank you, no, thank you," and they spun up a full-time research scientist position for myself, which I've been working at today. So just this year, I got promoted to senior quantitative ecologist, same science role, just slightly better pay, which is fun. But yeah, what is this like? How do you do quantitative science like at a zoo, right? That seems really strange. The Lincoln Park Zoo is quite unique in a lot of ways. We've got about 45 full-time science staff at the zoo, which is huge considering there's only about 250 full-time people working at the zoo. Um, we are probably in the top five, at least, in terms of conservation and science staff. And yeah, so it's really impressive. I would say that my job is I mean, it's so much fun, specifically because I get to focus most of my time on doing research instead of specifically on doing lots of other responsibilities you may find yourself having in academia, for example. Um, but yeah, I mean, I, for the most part, spend most of my time programming on computers, things like that. Um, like also said, my research is in biodiversity informatics. What that means is essentially, in a nutshell, I clean messy data sets. That is, of course, probably about 80% of anybody's work if you're really interested in working with big data, things like that. And then, while you're cleaning data, you need to know where it's going to, right? And what it's going to fit within. And so I also build lots of new statistical tools to answer basic and applied questions in ecology. All right. So with some of that stuff just about me out of the way, what are we going to be talking about today? So today we're going to be talking about UN, the Urban Wildlife Information Network, basically how we went from a single camera trapping study in Chicago to a global project today. And with this talk, like if we were all hanging out in person, I'd ask for everybody to give me a show of hands here and answer the question, do generalities exist in urban wildlife ecology? Since we're not in person, I'm just going to go ahead and guess. If we were raising our hands, maybe about half of you would raise them. I personally would say that yes, generalities do exist in urban wildlife ecology. Um, and one of those patterns that we tend to see is that if, say, we've got this regional species pool of nice colorful dots, and we've got an urban area and a quote unquote natural area. There's no such thing as really quote unquote natural anymore. Humans have kind of touched everything. But if we have urban and natural, right, and we go to a little habitat patch in those two different habitat types, often at the local scale in urban areas, you find fewer species at those habitat patches than you would in natural habitat patches for things like birds, mammals, things like that. Opposite pattern for plants, but we're not going to be talking about those today. And so why does this happen, you know? Well, part of the reason why this happens is that when you sort of pave paradise and put up a parking lot, you first increase the amount of isolation between habitat fragments. This is not uniformly bad. It can be good if there's colonization competition trade-offs between species, things like that. But one thing that is pretty uniformly bad for most species is that as you continue urbanizing areas, you're really reducing fragment size, things like that, which lots of species very, very negatively respond to, which in general results in there being fewer species in local habitat patches. When you combine that, of course, with a slew of just different mortality sources in urban areas, things like cars, things like cats, yeah, I mean, it could seem like that urban environments kind of spell doom and gloom for lots of animals. But of course, that is not the case, right? Like, I think that we've all seen really cool wildlife in cities. And part of the reason why that we do see lots of interesting wildlife in cities is because it's not just doom and gloom, right? There's lots and lots of opportunities if in cities for wildlife if they can adapt to the negative aspects of city life, right? So this can include things like anthropogenic food and water sources, and anthropogenic denning sites, protection from predators, even all that type of stuff, right? Um, and so that makes the field of urban ecology especially like really interesting to me because, at the end of the day, cities are built for and by people, but wildlife live here anyways, and they will continue to live beside us. And so, and they're responding exactly to what we're doing on the landscape. So I think that's just a really interesting system to study. Um, One thing that I think makes it difficult to sort of find generalities, especially in urban ecology, is essentially the fact that cities are not carbon copies of one another, right? Seattle is different from Chicago, is different from Austin, Texas, is different from so many different places. This is a figure that I made for a book chapter that my colleagues and I put together this last year. Briefly, I classified the dominant land cover class across a bunch of different cities throughout the United States and then clustered them. What you see up in the top right there are essentially cities that are predominantly agriculture. So those are in the Midwest, right? What you see on the left are predominantly forested cities. So places like the West Coast and the East Coast. And then in the American Southwest, you get mostly shrub, right? So we know that the dominant land cover classes are different, right? And that means there's bound to be different species in these areas. And that's going to complicate things a lot when you're trying to talk about general patterns that you see or try to understand findings in one city and apply them to your own, right? Um, Another way I like to think about sort of untackling this specific problem here is thinking about the spatial scale at which what's about the sources of variation that you may actually be interested in quantifying, right? I would say for the most most urban ecological studies focus on this very bottom scale here, looking at variation in occupancy, abundance, things like that within cities, and correlating it to things like green space type, human activity, housing density, stuff like that. Um, However, if we increase our scale and look at the differences, say, for example, between cities, like these two little stars right here, we could be looking at variation in the amount of built infrastructure between cities, cultural attitudes, human population, et cetera, et cetera. Take, for example, those two starred spots, Chicago, Illinois, and Indianapolis. Those are only about, 3 hours away from one another, but have very similar wildlife communities in a lot of respects, but are vastly, vastly different from one another. Chicago is about half the size, but has about 3 times the population. And of course, Chicagoans do things a little bit differently. We run our hot dogs through the garden, Chicago style hot dog up at top. Indy, of course, does not do that. Um, and then when we increase our spatial scale to variation between regions, we can, ask questions about climate and land cover. Take, for example, Seattle, Washington, forested and wet, right? If you compare the species that are there to Phoenix, Arizona, which is deserted and dry, and how species respond to urbanization between those two different cities, it's bound to be different. And I would argue the only way you can address questions about and answer questions about these three different scales is with big coordinated multi-city studies. And so that's what my coworkers and I at the Lincoln Park Zoo did. We started a research network basically, predominantly in the Midwest, with people that we had been hanging out and collaborating with for a few years, around 2016, 2017. And we don't really know how it happened, but it kind of snowballed from there. So we'd gone from about 5 to 7 different cities as part of the network in 2017 to today, 2024, when we're up to 60 different cities systematically collecting the same exact data as us worldwide, which is just awesome. It's super, super exciting to see this. I'm actually heading down to Bariloche, Argentina, down at the very, very south there in a couple months to go teach. Occupancy modeling and the like. So really looking forward to that. But yeah, UN is this big systematic multi-city collaboration. Um, and it's systematic because we use a common study design across our cities. And so with respect to camera traps, at least, this is not the only form of data that we're collecting across cities, but I'm only going to be talking about camera trapping today. We work with partners to sample wildlife along a gradient of urban intensity, so urban to rural areas. We work with them to select sites that follow that urbanization gradient very, very well. And we place camera traps for the most part in urban green space. And so this could include things like city parks, natural areas, cemeteries, golf courses. There's some private land there, things like that from time to time. But these four sort of different quote unquote site types cover most of where our cameras are located. And this is just two examples of what these two different transects look like for UN cities. I did not know there was another Manhattan, Kansas until they joined the network. It's the Little Apple, not the Big Apple, apparently. Um, but they're much, much, much smaller. Their transects only run about, I don't know, 5, 10 kilometers long. Whereas in Chicago, in order to get to sort of rural areas, we need to go at about 50 kilometers. Once we got data together from a few different cities, one question that I was specifically interested in, seemingly simple, right? Is that as cities become more densely populated, does how common a species is, what does the relative occupancy actually change? Is it in a densely populated city versus a non-densely populated city? So to visualize this a little bit, let's think about the x-axis here. We've got the average housing density of a city, and then on the y-axis is the average occupancy in a city. It could be that all of these points represent estimates for a single species in those different cities. It could be that we don't see any relationship, right? It could be, of course, there might be species that you see a very strong positive relationship. This might be pest species we really don't like, right? Things like bed bugs, things like rats. Um, but for the most part, I'm guessing for many different mammal species, especially, that we're likely to see a pattern that's like this, right? Um, Furthermore, can we actually bake into our statistical model a way to sort of quantify these specific patterns so we can actually make predictions into out of sampled areas, things like that. And the answer is, in a nutshell, yes, you can use cross-level interactions. I'm not going to go into this too deeply, but you've got covariates at the site level and at the city level. You include both, their interactions, some random effect terms, bada bing, bada boom, you're good to go. So this is what that sort of looks like when you do this. And this is a paper I published a few years back. Same exact X and Y axes here. So average housing density on the X, average occupancy on the Y axis. This is for Coyote. Um, the the dots, of course, are specific cities that contribute to data. And for Coyote, they don't really care. They occupy about 37% of habitat patches, regardless of how densely populated a city is. But there are some species that respond pretty darn strongly to this pattern here. Take, for example, the raccoon. Their occupancy precipitously declines as the average housing density of a city goes up. And so let's just go back to the Little Apple in Chicago. So Manhattan, Kansas, super, super small. Raccoon are basically everywhere there, whereas in Chicago, Illinois, there are only about half their sites. Um, but we can do more than this too. Instead of just looking at how common a species is, we can actually quantify how, average housing density in a city, things like that, can actually be used to predict where they're located in cities. And I just want to provide one small example of this. Um, this land cover map is actually you might be able to guess what it is, just because it's so close to you. This is Portland, Oregon. Portland is a UN partner, and they have an average housing density of about 840 houses per kilometer. Those black dots are the sites that they sample. When you compare that to Chicago, Illinois, we've got about 400 more houses per kilometer. All right, so if we look at this figure here, x-axis exactly same as everything else. The y-axis is essentially the response, or not response, the answer you'd get from your occupancy model with respect to how this specific species, raccoon, responds to site level housing density. So say, for example, if my Portland partners and I conducted independent studies, right? And we analyzed our data, including housing density as a covariate. Um, this model here predicts that I in Chicago would find a negative effect, and my partners here in Portland would maybe find a negative effect, but there might be a lot of uncertainty there. Who knows? What's super exciting about UN, of course, is that because it's a long-term study, we have independent data sets to validate this. And so I did pull some earlier this week. I did pull some data to refit this model, and this is what I got. So yeah, I mean, I'm pretty on the nose with this. This is, of course, a pretty cherry-picked example. I'm sure if I'd applied this to another city, it'd end up completely off. But it does definitely show that like we're both right here, right? There's not a specific like, well, Fidino et al. Found a negative effect, whereas we found maybe a marginally negative effect, maybe a positive effect, who knows? It's not that at all whatsoever. We're both right here. It's just that the context of our cities is different. To bring that back together, like if all UN partners analyze their data by themselves, we would find these big dissimilar species-specific responses to urbanization. But when we actually come together and work together, we collaborate, right? It allows us to see that these disagreements are explained by these big landscape scale differences between cities. And what's really great about that then is that it allows us to move away from like ecologist like it depends kind of conversation and moves us towards it depends because, right? It shows us that generality is possible when you actually understand the environmental context and how those differ among cities, which is really, really important. Um, with UN data, we could also use it to test a number of prevailing sort of different urban ecological hypotheses. One of those is the luxury effect. So it's this idea that species richness in cities is more often positively correlated to wealth, like per capita income. Very strong evidence of this in plants, and it's kind of goes in varying directions with other taxa. Um, so we decided to apply this to our UN data. The network had gotten much larger by the time we had started this project. About 20 different cities contributed data to this one. The model I used for this project is from Sutherland and al. They unfortunately misspelled the title of this paper, so I went ahead and fixed it for them here. If you've not read this paper, it's really fascinating. It's really great. I'll explain in a nutshell what this model does. So, UN data is, I mean, UN's not unique in this, but one thing that when I started working with UN data that I realized I needed to take account for was the fact that some species are just not available for sampling in some cities, right? So to take, for example, mule deer present throughout the western US, whereas Virginia opossum mostly present in the eastern US, right? And so this model adds another hierarchical level to your occupancy model to account for that, whether or not a species may be present for sampling within a city. And we can use that to estimate species richness within cities while accounting for that type of error. This model also can be used to share information among cities and species through its hierarchical structure. And then, of course, we can estimate occupancy at sites as a function of things like urban intensity and per capita income. And when we do that, and calculate species richness at all these different sites across UN, et cetera, et cetera. We found that we actually only had about 10 of these different cities. We've got cities on the y-axis here, and on the x-axis, we've got the correlation between species richness and and then either the income or urbanization gradient of a respective city. So the green that you see there is, of course, for income. And, and those vertical sort of black lines are median estimates, and those little distributions are 95% credible intervals. Um, Only about 10 of these 20 cities, we quantified a quote unquote luxury effect, so a positive correlation between income and species richness. But one thing that we did pretty much uniformly find across almost every single city was a very strong and negative relationship between species richness and urban intensity, urbanization, which isn't surprising, right? That confirms decades of urban ecological research. But it was kind of interesting to see that if we've got 20 different cities, maybe one of the biggest studies looking at the luxury effect to date, we only found it in about half of them. And that kind of leads us to the question, like, well, dang, like, why did we find it in only half of these cities, right? Like, why did that happen? Um, and so during the same time that we were working on this project, my buddy Chris Shell put out this absolute banger of a paper in Science. If you've not read it, I 100% suggest you look at it. And it really focuses on how like we've been spending so much of our time looking at things like income, and that's really just a symptom of so many different processes that are happening in cities. And we really need to start looking at how things like structural racism and classism can have these big bottom-up effects that can structure entire urban ecosystems, right? And so while Chris and I were having a beer together, we were chatting about this with the luxury effect and this paper, all that stuff together. And one thing that sort of came up from that, we decided to sort of look a little bit more into, was actually hidden within this word cloud in one of the figures that he put in there, and that's actually gentrification. Words and their definitions are important, right? When I say gentrification, I think everybody thinks up a different thing, right? Um, and because of that, I want to especially make certain that I take the time to define that well. So I'm just going to go ahead and define gentrification very explicitly here. I'm just going to read it off for everybody after I have a little drink. All right, and that is gentrification is the process of neighborhood change that includes both demographic and economic shifts in historically disinvested neighborhoods. So that's the important part. They're disinvested where wider, more educated, and higher income residents displaced under-resourced residents. Um, and so when we were thinking about like how could gentrification, for example, influence the composition of wildlife in cities, I initially wanted to shy away from trying to look at that with respect to how a whole slew of different species respond to gentrification, because I feel like it'd be really difficult to try to, I don't know, interpret what's going on there, right? Um, and so instead, I really wanted to look at some higher level um, sort of processes. And so I instead decided to focus on variation in alpha diversity, so species richness, and beta diversity, so variation in similarity between sites. And so let's take, for example, we've got two different sites, right? They could be one site's not gentrified, one site is gentrified. And these little shapes indicate species that are present in those areas. The filled in dots, filled in shapes mean that they're there. So it could be that species richness is the same, but they just have very distinct and very different communities. And so beta diversity would take a value of 1 here, but alpha diversity would be 0. Um, it could be that you lose a species and gain a few, right? And so we'd see a pretty big increase in beta diversity, not quite as high as distinct, but a big shift in alpha diversity. Um, we could instead, and this was my biggest guess of what would be actually happening, we'd have sort of nested subsets where you just have the same species, but more basically in gentrified areas. And then finally, it could be that there's just no difference, right? The null hypothesis. But I guess the question is though, like why Mason, do you want to understand if gentrification could influence the composition of wildlife, right? And to that, I would say, well, because if gentrification is actually tied to changes in sort of alpha diversity and beta diversity across a bunch of different cities, it actually helped provide some pretty a lot of evidence that there's aspects of urban nature that are just simply chronically inaccessible for some residents over others, right? Gentrification displaces a huge number of people, and it's actually not just one of those like it sucks sort of things, right? There are people die from from this actually. It's a pretty bad thing that occurs in cities. And in order to sort of uh, uh, you know, combat something like gentrification, if that's something you're interested in doing, you need to be able to talk about uh, its effect in a bunch of different ways. And speaking about it in terms of biodiversity is something that I felt that I would actually be well suited to do. So that's why I decided to start this project. Anyways, I digress, right? So to look at whether gentrification is actually associated to patterns of alpha and beta diversity. We got a whole bunch of UN data from 23 different US cities here. We had to reduce down to just the US because of census data, basically. But this is almost 1,000 different sites, almost 200,000 camera trap days between 2019 and 2021. Um, all right, so I'm not going to get into specifically how I quantify gentrification. It's just a whole bunch of spatial data analysis, pulling in census data, things like that. But this does follow that definition that I provided earlier. So these areas were disinvested in and were um, predominantly non-white, lower income, et cetera. And then they changed over the course of a decade, such that they became much more white, much more higher income, much more college educated, et cetera, et cetera. And so this is what it looks like for Chicago, Illinois. And so these are the gray census tracts are gentrified areas, and the brown census tracts are non-gentrified areas. The dots, of course, are all the sites that we sampled throughout Chicago. And then I essentially just calculated whether or not a single site was within 500 meters of a gentrifying census tract, and then had a binary variable in the model to determine whether or not a site was gentrified. So um, this modeling approach was actually probably the thing that took the longest for this project to get wrapped up. I first, of course, started with this Sutherland et al. Model. It's something that I'm probably going to be using with UN data for ever, if I had to guess. Who knows? But because I wanted to look at variation in alpha diversity and beta diversity, this model doesn't really get us there, right? You can look at the community average for the slope terms and things like that with the random effects, but that still doesn't quite get us there, I would say. And so I had to do something different. So to look at variation in alpha diversity, I first took the output from the occupancy model and estimated species richness, the expected species richness, as well as the uncertainty in that as well. So this is just what that looks like. On the x-axis, we have the expectation, so expected alpha diversity. On the y-axis, we have uncertainty in that estimate, so the standard deviation of that alpha diversity estimate. And from that, I took both of those things and actually fit that into a secondary model. I'm calling this like a meta-analytic model because it's kind of the same exact thing you do for a meta-analysis, except I'm not taking published research. I'm taking the results from a single study, from a single analysis, and just sort of chaining it into another analysis. This is kind of the same exact thing like you'd find in some of the applied hierarchical modeling and ecology books by Carey and Royal. So there's really nothing new here. It has an additional error term to sort of account for that uncertainty and the response variable, which is really cool. And then in the model, I included impervious cover, gentrification, and then the interaction between impervious cover and gentrification as well. And of course, we've got all of this hierarchical structure to the model to account for variation across cities and things like that. So city level random intercepts and slopes. Um, to look at variation in beta diversity, we kind of did the same exact thing. So we calculated pairwise differences. We calculated pairwise dissimilarity between sites within each city. And so on the x-axis, we have the expectation for beta diversity. On the y-axis, we have that standard deviation. And we see here, of course, that as you get sort of that. Oh, it doesn't matter. Actually, I'm going off on a tangent. I'm going to bring myself back. All right. For the next step, though, there really wasn't a specific statistical model that was around and really explained and developed that well to go from occupancy model to beta diversity as a function of covariates. And so that's sort of what took me a while working through this. What I ended up landing on was using generalized dissimilarity models because it uses a regression-based framework to do this. Um, but I had to spend a fair bit of time sort of going through a whole bunch of C++ code to convert it so I could actually write up my Bayesian model to do all of this instead. So that took me a tiny bit, but it sort of follows the idea of the alpha diversity model in spirit. So we've got this mean beta diversity as the response variable. We've got an additional error term in the model to account for that uncertainty in the estimate. And then we, of course, included impervious cover and gentrification as covariates in the model. And then we controlled for geographic distance between sites because that, of course, is going to be something that has an influence on beta diversity. And then city level intercepts and slopes. So what did we find? If we look at it at the species level, there was about 10 species that were more common in gentrified areas when impervious cover was low. And then there were about 3 species that were more common in just gentrified areas in general, things like woodchuck, raccoon, and I think cottontail rabbit. Um, but when we look at it with respect to alpha diversity, the answer is yes. The among city estimates are these black lines, and then the 95% credible intervals are the gray rectangles. And we got city specific estimates as blue dots. And we see here that this gentrification term is positive. The effect that impervious cover has is, of course, far more negative. So much greater effect there. And then the interaction between the two is also trending negative. And to interpret that is, of course, much easier to plot out these relationships to understand it. So let me show you what that looks like. So on average, across cities, gentrified parts of a city had about 1 to 2 more species than non-gentrified parts of a city. But this difference was actually much greater at lower levels of impervious cover. And you could really see this here on for Chicago, for example. So we've got on the x-axis impervious cover and on the y-axis alpha diversity. And if you're at about, say, 20% impervious cover for Chicago, you can see that there's about a difference of 5 to 6 species there. The reason why I think that we actually don't see a very negligible shift in species richness as impervious cover goes up is that as impervious cover goes up, you're just going to have far less habitable area for these species. And so it doesn't really matter if it gentrifies or not. There's just not much habitat potential there. Whereas in areas where there's far less impervious cover, there's at least potential for there to be habitat. And so that's going to be where it's going to have its greatest effect. The beta diversity results are really not that exciting. It was way more fun to figure out how to do the modeling than it was to look at the results for this one here, if I'm being honest. Gentrification, very negligible effects. It was mostly dominated by things like impervious cover and geographic distance between sites. But beta diversity was correlated to gentrification in some specific cities. So say, for example, in Metro LA, we did see beta diversity was about 1.2, 1.3 times greater in gentrified areas versus non-gentrified areas, whereas other cities that like Phoenix, Arizona, it's essentially really overlapping. While working on this project, one thing I was really trying to do was try to figure out how to visualize what this looks like at sort of the scale of the United States. And so one thing I decided to do was to put together a bivariate choropleth map. To do that, I essentially just calculated which cities had a greater alpha diversity effect versus a beta diversity effect, or what cities had a greater beta diversity effect than an alpha diversity effect, color coded them and plotted them out here. And we do get some kind of interesting sort of general pattern here, where we see, especially on the East Coast, we have cities that much more strongly respond to gentrification is more strongly associated to alpha diversity, whereas if you're on the West Coast or in more westerly cities, we see bigger shifts with respect to beta diversity, which is mostly driven by the fact that there was a very, very little to no shift in alpha diversity in those specific cities. So the question is why, right? Like I didn't want to see this pattern and then try to fit it with refit the model to quantify that, because that would kind of be like cherry picking the results there. So why do I think this is happening, right? Well, it could be that East Coast cities and more central US cities are more biotically homogenized. This has been found with birds, for example. Those cities are much older, so it could just be that they've had time for, all the species just to sort of move into those spots. I don't know. Um, West Coast cities, especially from 2019 to 2021, were subject to some really extreme weather events. There were some really extreme droughts, things like that. So it could be that during drought times that gentrified parts of the city may have benefited from additional water resources, something like that. I have no idea. But what it does show to me is that we just need additional independent data sets to answer this question. And what's exciting about UN, of course, is that it's this big long-term study, right? And so it's something that we can do with this. We just need to take a little bit of time to build up the data to do so. Um, but yeah, we found in general that impervious land cover had some of the strongest effects on alpha and beta diversity, and then gentrification really worked to modulate this pattern, right? It wasn't enough to sort of completely um, get rid of the negative effect of impervious land cover, but it was there to modulate that pattern at least a little bit. And that kind of brings me back to sort of this luxury effect study that we did too, right? We found this sort of to be true there as well. And so it's really showing that there's this like, gray part of the city, this impervious cover, the urbanization part of it. It's really strongly controlling things, but the social landscape of cities is actually having a pretty interesting effect too, which means it cannot be ignored. Um, Gentrification, like, why is it happening? My guess is that it's remediating local environmental disamenities, which could lead to improved environmental quality. But of course, it's imperative that we remember that gentrification is displacing people as well, right? So that means that there's aspects of urban nature that are less accessible to some residents. Um, So our results here demonstrate that gentrification, works on a pretty slowish timescale, 10, 20 years or so, but it has an influence on both humans, on the distribution of humans and wildlife, right? And because of that, because gentrification shapes where species are distributed in a city, it does also mean that gentrification shapes who has the opportunity to observe those species, right? And if we want to try to address these sort of environmental inequity issues, the best time to do it was decades ago, but the next time best time to do that is now. Um, so like, how do we actually address these types of problems, right? Um, Thankfully, I'm not the person that has to come up with those specific answers. I'm more just the statistician working on trying to identify some of these problems. But thankfully, there's a lot of really great literature out there that has some suggestions. So things like how land development policies should be prioritizing social equity. We really need to stop treating urban green space as a commodity for affluent white communities. And most importantly, we really need to start treating urban green space as an essential component of city maintenance instead of as an economic development strategy, which is what it's largely used like today. Um, but I would say that large-scale research networks like UN are really positioned to assist decision makers to understand these types of problems as they occur across cities and across scales, which can really help provide some valuable insights for building both equitable and biodiverse sort of cities. With that, I'm going to just share a few different things that are sort of on the horizon for myself, things to sort of look out for. So one thing I did not talk about today was this nifty R package my some of my buddies and I put together. So Brian Gerber, Kadambari Devarajan, and Zach Ferris and we published a paper earlier this year in Journal of Animal Ecology as a companion piece to this diel niche R package. Um, which essentially is a way to quantify a species' diel phenotype, like nocturnal, diurnal, crepuscular, etc., with data instead of just looking at a histogram of their activity patterns and kind of going off of vibes, which is what a lot of people have been doing. I just actually found out today that a paper that we've been working on for the last few years, where we applied that method to about almost 450 different mammal species worldwide, just got accepted. So probably early 2025. Look out in Science Advances for that. This is just one little result that I cherry picked to share here. I'm showing how as global human footprint increases, the nocturnality of a whole bunch of different species also increases. That is not available porcupine. That is North American porcupine. I just didn't have space for them. Um, Other things to be looking out for. I put together an R package for fitting autologistic occupancy models a while back, and I'm writing up a companion paper for that. Just about done with that. Should be submitting that next year. This R package, though, is 100% already available. You can find it on my GitHub. You can't fit this class of model in unmarked, so that's why I made it available. It's been really helpful for UN partners because I made it with them in mind because they have data that this would be well suited for. But lots of people have camera trap data or detection on detection data through time, and so this is potentially helpful to a lot of different people. Um, and then finally, UN is moving much more specifically towards ecological forecasting. This is one small example of predictions for where raccoon are located throughout Chicago, Illinois by census tracts. Um, if you're somebody that's really interested in ecological forecasting or developing automated systems for ecological forecasting, things like that, I would love to chat with you. I've got a lot of stuff cooked up right now, but it'd be great to have some more people in the kitchen. But yeah, it's been a fun thing to get to work on. With that, this slide here is already out of date. There's so many UN partners that I have to thank. But if you are interested, you can find me on Blue Sky. And then also, I'd just like to point out my personal website here. It's got all the papers that I've talked about today on there. And also, if you're somebody that does occupancy modeling, things like that, I've got a blog on that website that really showcases how to use a bunch of different classes of occupancy models, predominantly in a Bayesian framework, things like that, that people have found exceptionally helpful, I've found out over the years. So if that's something that you would find helpful, you should check it out as well. And with that, I didn't see any questions in the chat. So if people have questions for me now, I'm more than happy to take them.
Okay, thank you so much for such a great talk. Yeah, we're gonna take some questions. Feel free to submit your question in the Q&A box, or you can raise your hand. I can unmute people.
Thank you to whomever did the tiny clap. I saw it. If you don't have questions about the talk either, if you just want to ask me, it's cold here in Chicago. I don't know. Uh, all right, I did an excellent job, it seems like.
Yeah, it was like whenever I have a question in the previous slides, you right after you answered it. It's like, yeah.
With respect to that beta diversity modeling stuff, so the supplement material of that paper essentially is like a methods paper that's within the paper. And then if you look on my GitHub, the repo for that, there's all the data and code to completely recreate those analyses. I go out of my way to make certain that the README files are actually helpful so people can actually reuse things as needed. So if you're also struggling to think about like, what do I add to a README file for a project? Maybe look at some of those repos because they're great. Um, Thank you very much, Cole. Cole, I guess I'll read it out loud. Cole says, awesome talk and presentation. Thank you so much.
Okay, we do have a question in the Q&A.
Okay. Oh, I do not see it for some reason.
Oh, I
Would you mind
can,
just reading it and then I can,
yeah.
yeah.
So Noble says, can you talk a little bit about how you handled spatial autocorrelation in the trap detections along those gradients?
Um,
Yeah.
the long and short of it is that we don't. But we distance our cameras out by about 1 kilometer at a minimum to reduce spatial autocorrelation. There's times when I have had to account for spatial autocorrelation, and that's usually when I do go that route. That's usually with spatial smoothing splines, things like that. I found that to fit slightly better within occupancy models, at least in terms of making the model fit, you know, shorter than 1 month or something like that. Because when you add those like autoregressive like car models and things like that, things get really tricky. But yeah, um, we have also looked at using other classes of models like say for example the spot occupancy package from Jeff Dozer, which has like spatially varying coefficient models. But we haven't found it to be quite as useful with our data, maybe because they're so structured, right? We've got them all in a whole bunch of different cities, and it's not as uniformly placed throughout the landscape. But yeah,
And Noble said, fantastic talk. Really enjoyed the work you're doing.
cool.
Nice.
Thank you
Okay.
very much.
Got another comments questions. Sorry if I mispronounced the name. I'm really bad at this.
Oh,
Simone Simon. Yeah. Sorry.
oh,
Hi, I'm
actually,
actually, yeah,
I see it now. I see so from are you um the question from Simone?
yeah.
Okay. Um Yes, you are 100%. You made that figure, Simone. Awesome. Okay, so Simone says they're the artist scientists who work on that figure with Chris. Wonder if you've worked on any urban aquatic species. They tend to be drastically understudied. I really wish that we could be working more with urban aquatic species. In Chicago, we're fortunate enough, of course, to have the Shed Aquarium, and we're always talking about ways that we could potentially collaborate more. And we just really have not found a way to do that, especially at the scale of like, these big multi-city collaborations. Which is really disappointing because like in Chicago, for example, like we've been camera trapping there for the last 15 years, right? And we just detected our first North American river otter ever in like after millions of camera trap photos, right? And so like I think there's lots of really great opportunities to be studying like, either mammals that spend more time in the water or other species that just spend all their time in the water. Um, but yeah, 100, please email me. Yeah. Um, my email is on my website. So
Okay, awesome. Yeah, feel free to reach out by email or website or read the blog. Okay, I'm gonna close it up. So thank you everyone for coming to Quantitative Seminar today. This is our last session of the year, and I hope you enjoy the holiday and see you next quarter. Thank you.
thanks all.